Welcome back to Chapter 14. We're going to continue our discussion of antiviral drugs. At the end of the previous video, we looked at nucleoside analogs that prevent genome replication of DNA and RNA viruses. Our next group of drugs inhibit either entry into the cell or released from the host cell. For instance, amantadine and rimantadine, these block viral uncoating. So the virus needs to uncoat to infect the host cell. It needs to remove the envelope, if applicable, and get rid of the capsid in order to inject the nucleic acid into our host cell. But in this drug, if we block that uncoating, it's not able to infect us. However, these drugs are not used much anymore because of resistance. We also have oseltamivir, also known as Tamiflu, or zanamivir, more commonly known as Relenza. These block an enzyme called neuraminidase, and this prevents viral release from infected cells. So remember, viruses need to exit the host cell to attach to more host cells, and this prevents that from happening. We also have enfuvirtide, and it's misspelled in the book. So in the book, they have an extra I here that shouldn't be there. This one prevents HIV fusion with a host cell membrane. So all of these drugs reduce infection severity and shorten illness duration. When treating HIV, human immunodeficiency virus, we often combine multiple drugs to block different stages of viral replication. ART stands for antiretroviral therapy, whereas HAART or HAART stands for highly active antiretroviral therapy. And this was the more specific term used when treatments involved 3 or more drugs. As our drugs improve, modern treatments, often referred to as just ART or ART, often involve only maybe 1 or 2 drugs. So HAART is not used as commonly today as ART is. And these include a combination of reverse transcriptase inhibitors. Remember, HIV is an RNA virus, and they use their enzyme, reverse transcriptase, to convert the RNA to DNA before they integrate into our chromosome. So AZT is an example of a reverse transcriptase inhibitor we saw in the previous video. We also have protease inhibitors. This prevents the virus from maturing and being able to infect future host cells. So ritonavir is an example of a protease inhibitor. Integrase inhibitors prevent the viral DNA from integrating into our chromosome after they have already converted the RNA to DNA. And we also have fusion or entry inhibitors that prevent the virus from either binding to our cells or prevent the envelope of the virus from fusing with our cell membrane. So combination therapy prevents resistance and prolongs viral suppression. This figure shows the different targets of HIV therapy. HIV is an enveloped RNA virus, and one target is reverse transcriptase, which prevents viral RNA from being converted to DNA. Integrase inhibitors prevent the viral integration of their DNA into our host chromosome. We also have protease inhibitors that prevent maturation of the virus, so these viral particles will be unable to bind to and infect future cells. Fusion inhibitors prevent the virus from binding to the receptor on the host cells or prevent the envelope from fusing with our cell membranes. And similar to the previous table, our book also gives us a nice detailed table of antiviral drugs so you can see what is used to treat the different viral infections. But here we want to just focus for my class on the mechanism of action. So we had drugs that inhibit DNA or RNA synthesis. We had drugs that prevented the virus from being released, that inhibited uncoating or maturation of the virus. Don't worry too much about all of the names, although I would like you to know acyclovir for sure and AZT as well. This checkpoint asks, why is HIV difficult to treat with antivirals? HIV integrates its viral DNA into our host chromosome, making it a permanent part of infected cells. The virus also has a really high mutation rate, especially when it's reverse transcribing. So when it uses reverse transcriptase enzyme, this leads to mutations and quickly leads to drug resistance. HIV also hides in latent reservoirs like our resting T cells, which we'll learn more about later, and antivirals can't reach or act there. So treatment often requires some kind of combination therapy, targeting multiple viral processes like entry, reverse transcription, integration, or protease activity. So overall, it's hard to treat because it mutates rapidly, it integrates into our host chromosome, and uses our host cell machinery. So that really limits the drug targets and makes resistance, unfortunately, pretty common. All right, so to recap 14.4, the book asks us to explain the differences between modes of action of drugs that target fungi, protozoa, helminths, and viruses. So let's do that in the next few slides. We looked at antifungal drugs that disrupt ergosterol, cell walls, or mitosis in fungi. For our antiprotozoal drugs, they blocked metabolism, DNA synthesis, or heme detoxification. We saw our anti-parasitic worm or anti-helminthic drugs paralyzed or starved the worms. Antivirals interfere with viral enzymes or replication steps. So each drug class works by targeting features unique to its pathogen type, and that's the core idea of selective toxicity. And here are the same items, but placed in more detail within the table. You can see each type of pathogen. We have our eukaryotes, fungi, protozoa, helminths, and our acellular viruses, what they target, their modes of action, if they're selectively toxic, and example drugs in each category. Now moving forward to section 14.5, we're going to look at how microbes develop resistance to antimicrobial drugs. Drug resistance occurs when microbes survive exposure to an antimicrobial that would normally kill them, and this can happen through genetic mutations or gene transfer between bacteria. Over time, selective pressure from antibiotic overuse in medicine or agriculture, for instance, favors the survival of resistant strains, and this leads to treatment failures and persistent infections. This figure summarizes different mechanisms for drug resistance, how microbes resist the drugs that we try to use to kill them. So some microbes might have efflux pumps. These are pumps that do what they sound like. They are going to push the drug, such as the antibiotic, right out of the cell. We also have blocked penetration. Some microbes can alter their structures called porins, for instance, to prevent the drug from entering the cell in the first place. Other microbes will have enzymes that inactivate our drugs, or sometimes they even modify their own enzymes or own molecules such as DNA in order to prevent our drugs from binding to those sites. One major mechanism of resistance is enzymatic destruction or alteration of the drug. For example, we saw earlier that beta-lactamases, these are enzymes that break up the beta-lactam ring in penicillin, and that inactivates the drug. Other bacteria can modify aminoglycosides. Remember that aminoglycosides were ribosome inhibitors that bound to the 30S or small ribosomal subunit to cause mistranslation and faulty proteins. So if the bacteria can modify aminoglycosides, this will change their structures so they can no longer bind to the ribosomes. Overall, these enzymes make the antibiotic useless before it reaches the target. Some microbes can prevent the drug from entering the cell or simply push the drug out. So that strategy is either to block entry or again pump the drugs out. Gram-negative bacteria can change their porin channels to reduce uptake, or they can use efflux pumps to actively remove many different types of drugs. And because these pumps are nonspecific, they can lead to multi-drug resistance, where the bacteria will survive multiple antibiotic classes. This is commonly seen in gram-negative bacteria such as Pseudomonas aeruginosa or E. coli. The reason this can lead to multi-drug resistance is because these, again, efflux pumps are not specific to one particular type of antibiotic. So if you are taking different types of antibiotics, it can decrease the concentration within the cell of variety of many of those antibiotic drugs. Some bacteria can survive by altering the antibiotic's binding site. For instance, MRSA, methicillin-resistant Staphylococcus aureus, can change its penicillin binding proteins, and VRE, vancomycin-resistant Enterococcus, modifies the peptide chain in its cell wall. So in these cases, the drug can no longer recognize its target, so it becomes ineffective. Some microbes can go through target overproduction and bypass to overcome our drugs. So what's happening here is that cells like bacteria are producing an excess of the target enzyme or developing some kind of alternate metabolic pathway. For example, previously we talked about sulfa drugs, and sulfa drugs work by binding to the enzyme within the folic acid pathway and preventing PABA from being used, so the cells cannot make folic acid and thus they cannot make DNA or RNA. These bacteria, what they can do is overproduce PABA to overcome this problem. So if I look at what's happening, my substrate is PABA, para-aminobenzoic acid. My inhibitor is a sulfa drug. So usually when I have sulfa drug treatment, the sulfa drug is going to bind to that enzyme. But what happens if you overproduce PABA is now that it's much more likely that PABA will bind to the enzyme and create folic acid and ultimately DNA and RNA rather than the sulfa if it's not present in high enough concentrations. So this allows for bacterial growth even though the drug is present. Another type of pretty clever defense mechanism is target mimicry. And what happens here is that the bacteria are going to create decoy molecules that resemble the drug's real target. These will bind and neutralize the antibiotic or something similar. One example from our book is Mycobacterium tuberculosis, which creates MFPA. You don't have to worry about what it stands for, but if you're interested, it stands for Mycobacterium fluoroquinolone resistance protein. And what happens in this case is that this looks like DNA. So DNA gyrase will bind to this drug, and when they're bound together, fluoroquinolone is unable to bind to DNA gyrase. And usually what fluoroquinolone does is binds to DNA gyrase and prevents the DNA, remember the double-stranded DNA, prevents it from opening up to create a replication bubble and replicate the DNA. If the DNA gyrase is bound to MFPA, the drug again cannot bind to DNA gyrase, and when the drug concentrations have decreased eventually, now MFPA is released from DNA gyrase and cell replication can continue once again. This checkpoint asks for several mechanisms for drug resistance. So one is enzymatic inactivation or modification of the drug, where the bacteria produces enzymes that destroy or alter the antibiotic. For example, we have beta-lactamases that break down the beta-lactam ring in penicillins. The second one is prevention of drug uptake or increased efflux. So here bacteria are reducing the entry, such as altering their porin channels or pumping drugs out using efflux pumps. The third is target modification, where the drug's target site mutates so the antibiotic can't bind. For instance, we talked about MRSA, and they alter their penicillin binding proteins. The fourth one is target overproduction or bypass. This is when bacteria produce excess target enzyme or use some kind of alternative pathway. The example we talked about is actually sulfonamide resistance through overproduction of PABA, PABA. And last one is target mimicry. So here bacteria are making decoy molecules that resemble the drug target and protect the true target. So we just said Mycobacterium tuberculosis makes MFPA protein, which mimics DNA, and that protects DNA gyrase from the fluoroquinolone drugs. So overall, the bacteria resist drugs by destroying the drug, blocking entry, pumping it out, altering or bypassing targets, or by producing decoys. When multiple resistance genes accumulate, bacteria become multi-drug resistant organisms, MDRs, or also known as superbugs. Sometimes a single mechanism, like an efflux pump, can cause cross-resistance to many drugs. Because I mentioned earlier, efflux pumps are generally not specific, they pump all of them out. And what can also contribute to multi-drug resistance is incomplete prescriptions. So let's say you take 5 days of a medication instead of 10 days. Inappropriate antibiotic use can also accelerate this process. The escape pathogens are the key hospital superbugs that escape antibiotic treatment, and these include Enterococcus faecium, Staphylococcus aureus, Klebsiella pneumoniae, Acinetobacter baumannii, Pseudomonas aeruginosa, and different Enterobacter species. You may have heard of some of these major superbug examples. MRSA is resistant to all beta-lactams except some 5th generation cephalosporins that we saw earlier. VRE are vancomycin-resistant Enterococcus species. VRSA is vancomycin-resistant Staphylococcus aureus. ESBL, this is extended spectrum beta-lactamase producing E. coli and Klebsiella. And we also have CRE, carbapenem resistance, Enterobacteriaceae, so this is a family, and this includes gram-negative bacteria. Those are nearly untreatable, and there was a case study in our book as well. MDR-TB, this is multi-drug resistant Mycobacterium tuberculosis, which is resistant to, for instance, isoniazid and rifampin. So how does drug resistance lead to superbugs? Superbugs are bacterial strains that have acquired multiple resistance genes, and this makes them resistant to several or even all antibiotics. Resistance can arise through either spontaneous mutations in their chromosomal genes or horizontal gene transfer. We talked about this before through plasmids, transposons, those jumping genes, or conjugation, for instance, between bacteria. Overuse or misuse of antibiotics can also select for resistant survivors, allowing these resistant strains to multiply and spread. And over the time, this really leads to multi-drug resistant and extensively drug-resistant pathogens like MRSA, VRE, or CRE that we just saw. So superbugs emerge when bacteria accumulate multiple resistance mechanisms through mutation and gene sharing, surviving treatments that kill other bacteria, and spreading uncontrollably either in the clinical setting or community. To fight resistance, we sometimes use combination therapy, and our book gives us an example of Augmentin, which is a combination of amoxicillin and clavulanic acid. So clavulanic acid binds to and inactivates beta-lactamase enzymes in bacteria that are resistant to our penicillin drugs. So remember, beta-lactamase enzyme cleaves the beta-lactam ring, making our penicillin-type drugs inactive. But what clavulanic acid can do is, again, inhibit the enzyme. So now amoxicillin, which has this beta-lactam ring, can kill the bacterium. So again, this is an example of using more than one drug at a time to overcome resistance. Unfortunately, resistance genes can spread very quickly through different modes of horizontal gene transfer. So a while ago, we talked about the different types of horizontal gene transfer, including conjugation, where bacteria make a copy of their plasmid. This could be a resistance plasmid and sends it to the next cell. Transformation was when bacteria take up plasmids or DNA from the environment, and this could be resistance genes. And transduction was when a bacteriophage was involved, injecting nucleic acids, and all of these involve the transfer of genes between cells within the same generation. These cells can be unrelated species. This is why resistant infections can spread rapidly within hospitals. Unfortunately, routine antibiotic use in livestock promotes resistant bacteria that can enter our food and water supply. So these resistance genes can eventually transfer to pathogens that infect humans as well. So one way we can avoid the spread of antibiotic resistance or antimicrobial resistance is through veterinary oversight. What this means is that antibiotics should only be used in animals like livestock under the supervision and approval of a licensed veterinarian, and these antibiotics should not be used routinely or for non-medical purposes like growth promotion. Other ways to combat resistance include using antibiotics only when they're necessary, right? You should not be taking antibiotics for a viral infection or a fungal infection where it would be ineffective. We should always complete the entire prescription. So if the doctor prescribes a 10-day dose of an antibiotic, you don't want to stop after 3 days because you're feeling better, right? You always want to take the entire dose. We want to avoid sharing or saving these leftover antibiotics with others or saving them for future infections. We also want to make sure we practice infection control and surveillance. So what this means is that we have to identify which bacteria are becoming resistant and where the outbreaks are happening. We want to encourage research programs to support the development of new antibiotics or alternative therapies like phage therapy. So phage therapy is interesting. It's using phage, these are viruses that infect bacteria, to kill bacteria instead of using traditional antibiotics. All right, let's recap 14.5. So 1, explain the concept of drug resistance. Drug resistance is the ability of a microorganism to survive exposure to an antimicrobial agent that would normally kill it or normally inhibit its growth. In nature, microbes have always evolved resistance mechanisms to defend themselves against naturally produced antibiotics. But human use and misuse of antibiotics over time, such as overprescriptions, improper dosing, or incomplete treatments, have accelerated resistance, allowing these resistant strains to really thrive and spread. For the second one, describe how microorganisms develop or acquire drug resistance. We talked about 2 main genetic pathways. Mutation, this is random chromosomal mutations that come up during replication that can give resistance. And these mutations are passed on to future generations through vertical gene transfer. We also have our second method, which is horizontal gene transfer, where resistance genes are found on plasmids or maybe those jumping genes called transposons, and they can move between bacteria through conjugation, transformation, or transduction. For the third one, excuse me, describe the different mechanisms of antimicrobial drug resistance. Let's do this in the next slide. The third learning objective asked us to describe the different mechanisms of antimicrobial resistance, which we have here. So we saw that some microbes can modify the drug, such as having beta-lactamase, which inactivates our penicillin drugs. They can prevent the drug from being taken in or simply pump the drug out using those efflux pumps. Sometimes the microbe will modify the target, such as MRSA, that modifies its penicillin binding proteins. Or other microbes can bypass that part of the metabolic pathway, such as microbes that can resist our sulfa drugs by overproducing PABA. Others can mimic some kind of our drug target. For example, we talked about Mycobacterium tuberculosis, which makes MFPA that looks like DNA, so DNA gyrase will bind to that and therefore prevent fluoroquinolones from binding to DNA gyrase. That takes us to the end of our 4th video. In the next video, we're going to look at testing the effectiveness of our antimicrobial drugs.